Everybody. Our topic today is lithium neuroprotective. All right, can you see my slides? Excellent. All right, so our objectives for today, similar to some of the other topics that we've incorporated into the new curriculum for this year, this was one that has come up in discussion a few times. But recognizing that we have a brief didactic, this is one that definitely could take several different iterations. Since it's the first time we're offering this one, the focus for today is going to be to look at potential targets associated with neuroprotection, and then to describe lithium's actions on those neuroprotective targets. And to start with, I was trying to consider how best to tackle this and jump in, but I think it's important that we're all on the same page. So from a definition perspective, we're using neuroprotection as the numerous mechanisms and strategies that aim to protect the nervous system. And that's from injury, damage that could occur from toxic substances, from disease processes, from any of those type of things. So anything that's been found to be protective or can maintain neuronal health. And what we find is that there's a whole number of different causes of neuronal damage. We're going to look at these specifically because our focus today is on lithium. This is not an all-encompassing list. We are going to look at we're going to look at each of these individually in just a minute, both in terms of how it might be problematic for the neurons, as well as what lithium may do to that. So here are the 5 ones we're going to focus on. But I did, for full disclosure, we are focused more on the mechanistic side of what could potentially damage the neurons and how we could offset or reverse those effects. We are doing the mechanistic neural focus instead of the clinical focus because it's not a one-to-one translation. So just because there are mechanisms that have been demonstrated to damage the neurons, they're not always correlated with improvement in clinical functioning. So there seems to be quite a bit of inconsistency in the literature when we look at especially patients with bipolar disorder and their response to lithium. And because of time, which is why I have my little alarm clock up there, we're just going to focus on how lithium may be neuroprotective. So spoiler, it does have activities that are neuroprotective, but I was not able to do justice to the clinical aspect of that just within the time frame. So we're going to do foundational stuff today. So let's jump in. So excitotoxicity. The list of potential causes of neuronal damage was done just in alphabetical order. So it wasn't done in order of how much lithium plays a role in those. I thought, first of all, excitotoxicity was the first one that happened alphabetically, but this is also one of the ones that comes to mind very frequently when people think of something that might be toxic to the neurons. In this case, we're going to focus a little bit on glutamate. So recognizing that glutamate's one of our main excitatory neurotransmitters, it's been demonstrated that there is excessive glutamatergic tone in some patients with bipolar disorder. So this could be due to a change in the recognition and sensitivity of the glutamate system. So maybe there's an increase in the release of glutamate, potentially glutamate gets released to other stimuli. So it's a little bit more sensitive and more likely to be released than in other individuals. It's also been reported that in some cases, patients have a lower density of some of the glutamate receptors. So specifically the NMDA receptor, the N-methyl-D-aspartate receptor. So this receptor has been found to be in decreased, so those receptors have been endocytosed because they're extensively being stimulated by the release of glutamate. So interestingly, when they look at lithium's actions related to glutamate, it does a number of things. So it is able to inhibit some of the excitatory currents. So it has activity both presynaptically and postsynaptically. So presynaptically, it has been shown in some lab trials to decrease the release of glutamate. It also decreases the amount of neurotransmitters that are sometimes released as the result of an excitatory impulse. It also has activity on the postsynaptic sites, potentially changing the sensitivity and the response to an excitatory transmission. So we do see actions on both sides. They see more presynaptic activity at higher concentrations of lithium, but those that still would be therapeutic, so above that 1 milliequivalent per liter concentration. Other things that they found, so I mentioned that if glutamate is present relatively frequently, that we can see a downregulation in the NMDA receptors. So those receptors could be taken into a cell. They can have different components added to them. Phosphate often is added to them, and then they can be broken down. Lithium actually decreases the amount of phosphorylation of those receptors, as well as the deterioration of the receptors. So it's preserving the NMDA receptors that are present, potentially assisting with some of the downregulation, although the trials haven't been consistent that when patients are on lithium and they respond, that we see an increase in the density of those receptors. But potentially that could help with some of the expression in the overall management of the glutamate system. The other piece that lithium has been shown to impact the excitotoxicity component is an altered genetic expression. So, excuse me, there was a genome-wide association study that was done in patients with bipolar disorder. And they looked at those that were responsive to lithium as well as those that were not responsive to lithium. So they found 29 different single nucleotide polymorphisms. 19 of them were associated with regulatory elements. And when they looked at what do these regulatory elements, so it could have been a promoter or an enhancer of the genes, they wanted to know what was it. Well, they were both NMDA receptors as well as the AMPA glutamate receptors. So those seem to be correlated with patients that would respond to lithium. The other 10 SNPs were non-coding areas, and they haven't completely uncoded what they look at. So it does look like lithium can play a role in the receptor, essentially the translation and transcription and translation of the genes for the glutamate receptors. It may prevent some of the degradation of the NMDA receptors, and it may reduce the overall excitatory tone in some of those neurons. So trying to shift the balance and restore some balance in that particular system. For full disclosure, it also does have some inhibitory activities on the GABA receptor, but it is by far much more potent for the excitatory activity than it is for the inhibitory ones. So yes, there is still, it does diminish some of the inhibitory activity, but its effects on the excitatory side with glutamate have been much more pronounced. So next is the inflammatory system. So this one, there was a lot for this one, but lithium's actions are relatively small. So we're gonna do kind of a quick drive-by in the inflammation cascade. So in patients with bipolar disorder, it has been reported that there is dysregulation in the hypothalamic pituitary adrenal axis. So we can see that there's excessive corticotropin releasing factor, adrenal corticotropin hormone, and then some of those patients end up with higher cortisol levels. They also see a decreased sensitivity for some of those glucocorticoid receptors. And again, there may potentially be some downregulation because they're being stimulated on a regular basis. It's also been suggested that for some patients, there's an issue with the feedback loop in the HPA axis. For patients that are administered lithium, what they see is there is an activation of the HPA axis, which looks like, okay, if there's already dysregulation and we see some of that, how could that potentially be helpful? Well, it looks like it may reset some of that feedback loop dysfunction. So it's possible that not only does it encourage the HPA axis to be firing, but it's involved with some secondary messengers, and we'll get to them a little bit later, that may help reestablish the equilibrium and get this back on track. It also has some inhibitory actions on the corticotropins themselves. So lithium itself inhibits a number of enzymes, and that's where we're going to spend a little bit more time. Protein kinase C is one of them. So by inhibiting the activity of this particular enzyme, it actually may alter the overall feedback and expression in lower cortisol levels. So that enzyme is involved in the middle of the process, and also may change some of the regulatory proteins that are involved. So specifically cyclic AMP response binding protein. So that regulatory protein is involved in transcription and may really be cranking out a number of the hormones. So by inhibiting that, you reduce that activity, so you're not creating quite as many of those hormones, including the cortisol. So it looks like it's involved, and yes, it increases the activity of that, but it may actually reset the entire system. Again, not one of the main activities, but maybe secondary and something that's supportive to assist with some of the patients. These next couple areas tend to be where more of the focus is. So the mitochondrial dysfunction, now this is not a complete list of all of the functions of the mitochondria, but for our purposes, some of the activity that we see from the mitochondria include apoptosis, roles in regulating the intracellular calcium levels, which means that they may be intricately involved in intracellular signaling, as well as the release of neurotransmitters, and neuroplasticity. So potentially the management and maintenance of some of the neurons, as well as their ability to adjust, especially to insult. When there is dysfunction, it can be problematic, and it has been associated with a number of neurodegenerative diseases. And some of the mechanisms related to that are abnormal oxidative metabolism. So they're not able to get rid of those oxidative radicals that could potentially damage the cell itself, and damage to the DNA. So this damage in particular may cause an increase or decrease in expression of a number of proteins. These may have minimal role for the cells. These may play a massive role for the cells. The other thing that we see is that the telomeres, so those little caps that are on the end of the DNA, are shortened extensively in some patients that have mitochondrial dysfunction. And that may actually result in mutations in what gets transcribed from the DNA. So how can lithium play a role? So lithium's been associated with enhanced antioxidant production. So it decreases lipid peroxidation levels. So it can get involved in technically decrease some of the oxidative radicals that we see. It also has been associated with higher concentrations of antioxidant enzymes. So one of the main ones is the superoxide dismutase. So decreasing some of the oxidative components and increasing some of the antioxidant enzymes that are going to form more antioxidants can help reestablish and get rid of, if there is dysfunction, some of those oxidative radicals that can cause damage. It improves cellular respiration, and it stabilizes the mitochondrial membrane. So if the mitochondria are functioning properly, they actually can increase the word just went totally out of my head, the energy source, the ATP that's available for the cell. So it really has the energy that it needs to be able to carry out the functions that are necessary, especially if it's responding to stress or some other type of toxin. Lithium also seems to assist with balancing an apoptotic protein ratio. So this was a very interesting study that was done. They looked at a number of different factors, but they were looking specifically at lithium and valproic acid derivatives. And they wanted to see their role in a number of different cellular functions. And one of them was, this is the wrong one, but it's a very similar graph. I'll change that for you, Kay. Was supposed to be B, not C. So the one that I will show you looks very similar, but has concentrations of BCL as well as BAX. So BCL is actually an anti-apoptotic protein, and BAX is one of the pro-apoptotic proteins. So what they find is when there is stress to the neurons, the mitochondria, if there is dysfunction, start to produce more of the BAX protein, which will encourage the cell to undergo apoptosis or cell death. By having lithium or a valproic acid on board, they actually see that the BCL, the anti-apoptotic protein, comes back into balance. So it's not quite as diminished as it is in patients who don't have lithium. Now, in this case, they did not use, this was an animal model, but they didn't use animal models of bipolar disorder. They actually used methamphetamine as the stressor, as the aggressor. So compared to controls that were not exposed to anything, the balance of that ratio, there was a significant amount more of the pro-apoptotic protein in those individuals that were exposed to methamphetamine, but it was not as significant in those that were given lithium or the valproic acid derivatives. So it helped preserve some of that function. It also encouraged the genetic expression of a number of different proteins, including that the anti-apoptotic protein, the BCL. So it's involved in a number of different processes related to the mitochondria to help keep those neurons healthy. Other things it attempts to keep healthy, so we see a number of neural proteins that could be incredibly problematic. So in some cases, and this one's going to focus, most of it's been on bipolar disorder, but this is interesting, and it's more for the Alzheimer's disease patients. So they've been looking at treatment with very low doses of lithium as a prevention for the development of Alzheimer's disease. Now, some of the reasons that they've been doing that is that when patients are given lithium, we see increased amounts of brain-derived neurotrophic factor. So BDNF in both bipolar patients as well as those with Alzheimer's disease can really promote neuronal growth, keep those connections, and help with neuronal plasticity. So it can help with both of those. The other thing it's able to do, which is a little bit more unique for the Alzheimer's population, is those tau proteins. When some of the tau proteins are phosphorylated, if they have the phosphate, excuse me, attached to them, that's where they may start to bunch together and cause some accumulation, which may be some of the early precursors to the development of Alzheimer's disease. They actually see a diminishment in that phosphorylation of the tau proteins by lithium. Now, this has not been demonstrated to, if we give patients low-dose lithium early on, to completely stop the development, but they are looking at this as potentially being an option to help in some high-risk patients, and there are inconsistent reports on whether or not that's been beneficial. The other thing that it appears to do is lithium increases the autophagy activity. So it encourages those neurons to actually get rid of anything that is misfunctioning. So if there is a protein that's malfunctioning because it's got extra phosphate, if there's tau proteins that are accumulating, if there are other things that really aren't working the way that they're supposed to, it encourages the neurons to clean those out. Now, where that seems to be correlated to bipolar disorder is there are reports in adult patients who've been stabilized long-term on lithium that they have a preservation of their gray matter. So in some patients with bipolar disorder who've been relatively uncontrolled compared to a population that doesn't have bipolar disorder, they can see a reduction in the overall amount of gray matter. in patients that had responded to lithium, we can actually see a preservation where they're closer to the amount of gray matter that the patients without bipolar disorder have. And one of the suggested theories is because of this. They're able to get rid of any inflammatory activity, inflammatory agents, including oxidative radicals. They're able to get rid of anything that's not really functioning. So they're really cleaning out those neurons to be able to preserve as much neuronal function as possible. So interesting caveat, and a little bit beyond our bipolar population. The last section is really the one that has the most attention for lithium, and that's its activity with the neural enzymes. So one of the ways that it appears that lithium interacts with neural enzymes is because of its relationship to, we usually think of lithium and sodium, but its relationship to magnesium. So if you look at the picture here on the left-hand side, the lithium and the magnesium are relatively similar size. So even though lithium has a charge of 1 and magnesium has a charge of 2, because they are of similar size with the positive charge as a cation, lithium can be mistaken for magnesium as well as for lithium. That seems to be one of the ways that they believe it's able to displace magnesium, which serves as a cofactor for a number of different enzymes. We're going to spend a little bit of time on the next slide talking about the glycogen synthase kinase, so the GSK3 beta. That seems to be the main mechanism by which they think lithium may be neuroprotective, but it has other activity as well. So both the inositol monophosphatase and the inositol polyphosphate, so these two enzymes are intricately involved in cell signaling. So they're involved in cell growth, apoptosis, cell migration, potentially some of that endocytosis that happens with some of the enzymes. So if they're not functioning properly by inhibiting some of those enzymes, we may be able to re-establish the balance so that we don't get unnecessary cell apoptosis. Cells are able to grow and clean, as well as to migrate and have some more of that neuroplasticity. Other activities where lithium seems to compete with magnesium is with the AKT beta arrestin. So this particular enzyme also is involved in the signaling complex, and this is tends to be most prominently seen in some of the dopamine neurons. So if they can re-establish the activity with some of the dopaminergic neurons, it can actually assist in some subtypes of the bipolar disorder. So not all of the patients that have bipolar disorder have dysfunction in their dopaminergic neurons, and that's where some of that clinical correlation becomes a little bit sticky, because even though lithium may consistently have activity here, it doesn't always result in improvement in symptoms long term. The last one is the protein phosphatase 2. So this particular enzyme is involved in intracellular transport, as well as transcription and translation in DNA repair. So as we mentioned, if there's mitochondrial dysfunction, if we have a number of oxidative radicals, we may need to do quite a bit of repair for the DNA. This particular enzyme, if it's not functioning properly, there may not be enough of the enzymes available to be able to clean those things up, resulting in neuronal damage. So by inhibiting, especially if there's excessive activity, then we're not going to be constantly attacking or trying to transcribe a number of potentially unnecessary hormones, proteins, other components. So it'll help reestablish some of the equilibrium. This one in particular, though, is being looked at. They haven't found a consistent signal with bipolar disorder, but they are looking at it for Huntington's disease. They're looking at it for ALS and Parkinson's disease. So they're looking at it more potentially for some of the other neurodegenerative disorders. It hasn't had as clear of a link with bipolar disorder. But the GSK3 beta has. So there's two different glycogen synthase kinase enzymes. So there's alpha and beta. Beta is most prominent in the brain. it's involved with a number of different processes. So signaling, transcription, translation, apoptosis. So it seems to be involved in a number of different processes. Lithium actually can inhibit its activity in two different ways. So it can have direct inhibition, it binds where magnesium is supposed to bind at the catalytic core and reversibly inhibits any activity of the enzyme. It also has some indirect activity where it can cause phosphorylation of the enzyme, which results in a conformational change where it then can't bind its substrates, and it essentially is, again, reversibly inhibited. What this can do is prevent any type of apoptosis, and it's been shown to enhance synaptic plasticity. So when they reduce the activity of this particular enzyme, they actually can see the neurons go back to being able to establish new connections, even if they've been damaged due to toxins or a potential disease process. Now, this has not consistently been demonstrated to have a correlation with improvement in the clinical symptoms of bipolar disorder. There are a number of different factors that are involved. Some of it have to do with environmental factors, some of it have to do with genetics, but this does seem to be one of the indicators for if there's excessive GSK3 beta activity. Those patients do tend to respond to lithium. They may not be able to tolerate it long term, but it may be one of the indicators for that would be a good patient for this particular medication. It also is suggested that this may be why we see both. The other thing that can happen with the inhibition is remyelination. So we actually can see an increase in the white matter in patients who have responded and have been on lithium long-term as well. So that was a quick drive-by of the 5 potential areas for neuronal damage and how lithium actually may play a role in all 5 of them. So to wrap it up, since there are a number of different ways that this could happen, lithium is involved with most of these, but it's not consistently correlated to clinical application. So it does have some neuroprotective effects, but it's not going to be a quick, clean advantage for absolutely every patient. And not every patient's going to have that potential benefit, that neuroprotection that's as obvious. With that, I will turn it back over.